true titan in the field of computer science, Dr. Knight is widely recognized as the godfather of synthetic biology. His groundbreaking work has revolutionized synthetic biology field, and his co-founding the world-renowned Ginkgo Bioworks is a proof of his visionary leadership. So to begin this session, we'd like to invite our two esteemed panelists to share their perspectives.
My background is as a computer programmer, a computer designer, a semiconductor designer, and a professor teaching all of those things at MIT. I spent 40 years at MIT. I got all of my degrees at MIT. Joined the faculty in about 1983. And during that time, I became quite adept at building computer systems with whatever technology was out there. And so I started thinking in about 1990, what does the next technology look like? What is the technology that's going to overtake semiconductors in building the next set of really important things that are going to happen in the world? One way I started thinking about this was to say, all right, the core technology which we need is not building better lithography or better polishing or better etching. It's building better chemistry, because the chemists are the people who are able to put atoms precisely where they want them. I can build a chemical compound and every one of them that gets created is identical. They're not different, there's no statistics involved, they're all the same. And then you ask yourself, what's the best chemistry? And the best chemistry is biochemistry. And so somewhere in the early 1990s, I decided that if I was going to be technically relevant in the next generation, I needed to know about biochemistry, and I needed to know it in depth. And I essentially went back to school and became a biochemistry and a biology graduate student. And somewhere around 1996 or so, I was on advisory panels for the US government, notably DARPA, the Defense Advanced Research Projects Agency, the people who brought you the internet and GPS. They run advisory panels every summer, and I was tasked with developing a plan for what should be on that next advisory panel. What their project should be. And I proposed one that we called cellular engineering. And I got them excited about the possibility of building cells that did things in the real world, that built things that we're now building on a routine basis. And they funded my laboratory at MIT. I set up a microbiology lab, a BSL-1 lab at MIT in the computer science department. In about 2004, we established the very first iGEM program came out of my lab at MIT. That has gone on to grow to be a worldwide competition happening every year in Paris. It has educated probably something like 30,000 or 40,000 students over the years. Many of them are now faculty in synthetic biology at universities all across the world.